Some patterns only emerge when you read all tablets together, and some questions require more than unsupervised discovery. They need a specific outcome to predict. Jack all 3 tablets into a 3-dimensional Q, a tensor. Patients along one axis, features along another, omics type along the third. Tensor decomposition breaks this Q into a sum of simple components. Each component is a rank 1 tensor, a pattern that spans all 3 dimensions simultaneously. This captures interactions between omics layers that matrix methods like MOFA+ would miss. If a gene, its protein product, and a downstream metabolite all co-vary across patients, tensor decomposition finds the 3-way interaction. DIABLO is supervised integration. You know which patients responded to the drug. Now find the multi-omics signature that predicts response. It's like searching the tablets for words that appear together specifically in the passages describing victory. DIABLO uses sparse partial least squares to select a small number of features from each omics layer that jointly predict the outcome. The result is an integrated biomarker panel, a combination of genes, proteins, and metabolites that together predict treatment response better than any single layer alone. Weighted nearest neighbors comes from the single-cell world. Build separate neighbor graphs for each data type, RNA neighbors, protein neighbors, chromatin neighbors. Then for each individual cell, weight the graphs by how informative each layer is for that cell. Some cells are best defined by their RNA profile. Others are best defined by their surface proteins. 1 adapts per cell, giving each layer exactly as much influence as it deserves. This powers the multimodal analysis in Seurat version 4 and 5. How do you know the integration worked? Pre-checks. First, biological validation. Do the integrated clusters correspond to known cell types or disease subtypes? Second, classification accuracy. Does the integrated signature predict outcomes better than single omics? Third, silhouette scores. Are the integrated clusters tighter and better separated than single omics clusters? If integration doesn't improve on any of these, you may be adding noise, not signal. Integration isn't always better. If one omics layer is noisy, say your metabolomics had poor quality control, integrating it with clean transcriptomics will drag the signal down. Start by evaluating each layer independently. If one layer already gives excellent classification or clustering, integration may not help. And if your sample sizes are very different across layers, 10,000 cells with RNA, but only 500 with protein, the small sample layer dominates the integration artifacts. Integrate deliberately, not by default. 6 methods for reading the tablets together. GIF for paired translation. MOFA+ for shared themes. NEF for consensus networks. Tensor decomposition for 3-way interactions. DIABLO for supervised multi-omics signatures. 1 for per cell adaptive weighting. And the discipline to ask, is integration actually helping? Every multi-omics study should try at least 2 methods and validate the results biologically.